This module will give an overview about proteins that are involved in the transport and storage of oxygen and dioxygen activation. In this regard, we will learn about the natural proteins that carry dioxygen in living systems, understand the role of myoglobin and hemoglobin in carrying dioxygen in mammals, and the role of hemerythrin in dioxygen transport in marine invertebrates. We will also study various enzymes which act as activators of dioxygen for the inclusion of oxygen atoms in biologically important organic substrates. Most organisms require molecular oxygen for their survival. For some small animals and plants, where the surface to volume ratio is large, an adequate supply of dioxygen can be obtained from simple diffusion across cell membranes. The dioxygen may be extracted from air or water, and for plants that produce dioxygen in photosynthesis, it is also available endogenously. For other organisms, particularly those with non-passive lifestyles, diffusion does not supply sufficient dioxygen for respiration. Therefore, in these organisms, specialized molecules for the transport and storage of oxygen are necessary. These functions are carried out by a number of well-known iron and copper containing species which occur in the blood. These proteins are listed in the table. A good oxygen carrier should bind oxygen at high pressure, should not oxidize cellular components, and should give up oxygen on demand. Both heme and non-heme iron proteins are involved in oxygen transport and storage. Heme is a cofactor consisting of ferrocene contained in the center of a large heterocyclic organic ring called porphyrin. Porphyrin is made up of 4 pyrrolic groups joined together by methine bridges. Heme oxygen carriers are responsible for red color of human blood. They absorb 1 mole of oxygen per mole of ferrous, while the blue pigment of crab blood, hemocyanin, absorbs 1 mole of oxygen for every 2 moles of cuprocyanins. Certain marine worms have a violet color, which is due to presence of non-heme iron protein hemerythrin. Thus, myoglobin, hemoglobin, hemerythrin and hemocyanin all use a transition metal complex to transport or store oxygen. In the following sections, each of these and the 2 enzymes, dioxygenase and methane monooxygenase, which are involved in the activation of oxygen for incorporation in biological molecules, will be discussed. Myoglobin is a relatively small protein that contains 150 amino acids. The functional unit of myoglobin is an iron porphyrin complex that is embedded in the protein, as you can see in the figure. In myoglobin, the heme iron is 5-coordinated to a single histidine imidazole ligand from the protein called the proximal histidine, in addition to the 4 nitrogen atoms of the porphyrin. A second histidine imidazole called the distal histidine is located on the other side of the heme group, too far from the iron to be bonded to it. Consequently, the iron atom has a vacant coordination site, which is where dioxygen binds. In deoxymyoglobin, the iron is 5-coordinate and high spin. Because high spin ferrous is too large to fit into the hole in the center of the porphyrin, it is about 60 picometer above the plane of the porphyrin. When oxygen binds to deoxymyoglobin to form oxymyoglobin, the iron is converted from the 5-coordinate high spin to the 6-coordinate low spin. Because low spin ferrous is smaller than high spin ferrous, the iron atom now moves into the plane of the porphyrin ring to form an octahedral complex, as you can see in the figure. The oxygen pressure at which half of the molecules in a solution of myoglobin are bound to oxygen is about 1 millimeter mercury or 1.3 into 10 to the power minus 3 atmosphere. An important point to note is that a vacant coordination site at a metal center in a protein usually indicates that a small molecule will bind to the metal ion, whereas a coordinatively saturated metal center is usually involved in an electron transfer. Hemoglobin consists of two subunits of 141 amino acids and two subunits of 146 amino acids, both similar to myoglobin. It is called a tetramer because of its 4 subunits, but the framework of each subunit is same as that for myoglobin. Hemoglobin is not simply a supermyoglobin that can carry 4 oxygen molecules simultaneously, one per heme group. The shape of the oxygen binding curve of myoglobin is hyperbolic. In contrast, the oxygen binding curve of hemoglobin is S-shaped. The curve for myoglobin can be described by a simple equilibrium between the deoxy and oxymyoglobin. But the S-shaped curve for hemoglobin can be described only in terms of a cooperative interaction between the 4 hemes. When oxygen binds to the iron complex, it causes the iron atom to move back towards the center of the plane of the porphyrin ring. At the same time, the imidazole side chain of the histidine residue interacting at the other pole of the iron is pulled towards the porphyrin ring. This interaction induces a strain in the protein helix containing the histidine as it moves nearer to the iron atom. This strain is transmitted to the remaining 3 monomers in the tetramer, where it induces a similar conformational change in the other heme site, such that binding of oxygen to these sites becomes easier. In the tetrameric form of normal adult hemoglobin, the binding of oxygen is thus a cooperative process. The binding affinity of hemoglobin for oxygen is increased by the oxygen saturation of the molecule, with the first oxygens bound influencing the shape of the binding sites for the next oxygens, in a way favorable for binding. This positive cooperative binding is achieved through steric conformational changes of the hemoglobin protein complex, that is, when one subunit protein in hemoglobin becomes oxygenated, a conformational or structural change in the whole complex is initiated, causing the other subunits to gain an increased affinity for oxygen. As shown in the oxygen binding curves, at low partial pressure of oxygen, the affinity of deoxyhemoglobin for oxygen is substantially lower than that of myoglobin, whereas at high partial pressure of oxygen, the two proteins have comparable oxygen affinities. The physiological consequences of the unusual S-shaped oxygen binding curve of hemoglobin are enormous. In the lungs, where partial pressure of oxygen is highest, the high oxygen affinity of deoxyhemoglobin allows it to be completely loaded with dioxygen. In the tissues, however, where the partial pressure of oxygen is much lower, the decreased oxygen affinity of hemoglobin allows it to release oxygen, resulting in a net transfer of oxygen to myoglobin. Function of protein part is to provide a crevice which keeps heme from getting oxidized. In the absence of the protein, ferrous will get converted to ferric and hemoglobin myoglobin would become inactive. Heme is buried in the environment of the protein so that oxygen binding does not result in oxidation. Oxygen is not unique in its ability to bind to a ferrous heme complex. Small molecules such as carbon monoxide and nitric oxide bind to deoxymyoglobin, hemoglobin, even more tightly than does dioxygen. The interaction of the heme iron with oxygen and other diatomic molecules involves the transfer of electron density from the filled T2J orbitals of the low spin d6 ferrous ion to the empty pi star orbitals of the ligand. In the case of the ferrous dioxygen interaction, the transfer of electron density is so great that the iron oxygen unit can be described as low spin ferric and superoxide anion O2-. We can therefore represent the binding of dioxygen to deoxyhemoglobin and its release as a reversible redox reaction. As shown in the figure, the iron oxygen unit is bent with an iron OO angle of about 130 degrees. Because the pi star orbitals in CO are empty and those in NO are singly occupied, these ligands interact more strongly with ferrous than does oxygen, in which the pi star orbitals of the neutral ligand are doubly occupied. Although carbon monoxide has a much greater affinity for ferrous heme than does oxygen by a factor of about 25,000, the affinity of carbon monoxide for deoxyhemoglobin is only about 200 times greater than that of oxygen, which suggests that something in the protein is decreasing its affinity for carbon monoxide by a factor of about 100. Both CO and NO bind to ferrous hemes in a linear fashion, with a iron CO or iron NO angle of about 180 degree, and the difference in the preferred geometry of oxygen and carbon monoxide provides a plausible explanation for the difference in the affinities. If we look at the figure, we will see that the imidazole group of the distal histidine is located precisely where the oxygen atom of bound carbon monoxide would be if the iron CO units were linear. Consequently, CO cannot bind to the heme in a linear fashion. Instead, it is forced to bind in a bent mode that is similar to the preferred structure for iron oxygen unit. This results in a significant decrease in the affinity of the heme for carbon monoxide, while leaving the oxygen affinity unchanged, which is important because carbon monoxide is produced continuously in the body by degradation of the porphyrin ligand. Under normal conditions, carbon monoxide occupies approximately 1% of the heme sites in hemoglobin and myoglobin. If the affinity of hemoglobin and myoglobin for carbon monoxide were 100 times greater due to the absence of the distal histidine, essentially 100% of the heme sites would be occupied by CO and no oxygen could be transported to the tissues. Severe carbon monoxide poisoning, which is frequently fatal, has exactly the same effect. Thus, the primary function of the distal histidine appears to be decrease the carbon monoxide affinity of hemoglobin and myoglobin to avoid self-poisoning by CO. Hemerythrin is used to transport dioxygen in a variety of marine invertebrates. It is an octamer, that is, it has 8 subunits, with each subunit containing 2 iron atoms and binding 1 mole of dioxygen. These invertebrates also contain a monomeric form of hemerythrin that is located in the tissues analogous to myoglobin. The binding of oxygen to hemerythrin and its release can be described by the reaction. where HO2- ligand is the hydroperoxide anion. Ferrous plus oxygen plus H plus combine reversibly to give ferric O2H. Thus, oxygen binding is accompanied by the transfer of 2 electrons, one from each ferrous and a proton to oxygen. Hemocyanin is used for oxygen transport in many arthropods like spiders, crabs, lobsters and centipedes, and in mollusks like shellfish, octopi and squid. It is responsible for the bluish-green color of the blood. The protein is a polymer of subunits that each contain 2 copper atoms. Deoxyhemocyanin contains 2 cuprous ions per subunit and is colorless, whereas oxyhemocyanin contains 2 cupric ions and is bright blue. As with hemerythrin, the binding and release of dioxygen corresponds to a 2-electron reaction. Although hemocyanin and hemerythrin perform the same basic function as hemoglobin, these proteins are not interchangeable. In fact, hemocyanin is so foreign to humans that it is one of the major factors responsible for the common allergies to shellfish. Many of the enzymes involved in biological reactions of oxygen contain metal centers with structures that are similar to those used for dioxygen transport. In this section, we will briefly describe 2 of the most important examples, namely dioxygenases and methane monooxygenase, which act as activators of oxygen for the inclusion of oxygen atoms in biologically important organic substrates. As these reactions involve change in the oxidation state of the metal, so these are redox enzymes. Dioxygenases are enzymes that insert both oxygen atoms of oxygen into an organic molecule. In humans, dioxygenases are responsible for cross-linking collagen in connective tissue and for synthesizing complex organic molecules called prostaglandins, which trigger inflammation and immune reactions. Iron is by far the most common metal in dioxygenases and the target of the most commonly used drug in the world, aspirin, is an iron enzyme that synthesizes a specific prostaglandin. Aspirin inhibits this enzyme by binding to the iron atom at the active site, which prevents oxygen from binding. Iron-containing dioxygenases can be subdivided into 3 classes on the basis of how iron is incorporated into the active site. Those employing a mononuclear iron center, those containing a risk 2 iron 2 sulfur cluster, and those utilizing a heme prosthetic group. Methane monooxygenase or MMO catalyzes the conversion of methane to methanol. The enzyme is a monooxygenase because only 1 atom of dioxygen is inserted into an organic molecule, while the other is reduced to water. Because methane is the major component of natural gas, there is an enormous interest in using this reaction to convert methane to liquid fuel methanol. That is much more convenient to ship and store. Because the CH bond in methane is one of the strongest CH bonds known, however, an extraordinarily powerful oxidant is needed for this reaction. The active site of methane monooxygenase is shown in the figure, and we can see that it contains 2 iron atoms that bind dioxygen. But the details of how the bound dioxygen is converted to such a potent oxidant still remain unclear. Let us now summarize what we have learnt in this module. Nature has devised perfect molecules in the form of hemoglobin, hemerythrin and hemocyanin, which contain heme iron, non-heme iron and copper respectively, for the transport of oxygen with the right combination of structural features for easy uptake and release of dioxygen in the right conditions without oxidation of the metal center. Myoglobin, which contains heme iron, has the perfect structural features to act as a storage rather than transport protein for dioxygen. These proteins are non-redox proteins. That is, there is no change in the oxidation state of metal during the transport or storage of oxygen. On the other hand, dioxygenase and methane monooxygenase act as activators of molecular oxygen for the inclusion of 1 or 2 atoms of oxygen respectively in an organic substrate. This process involves a change in the oxidation state of the metal, and hence these are redox proteins. Thus, from these examples, we understand that transition metals can be redox inactive or active depending upon the coordinated ligands and the surrounding protein environment. And these features account for their wide availability in the biological systems.